Hello class, today we're going to be working on the mitosis and meiosis. This is pronounced meiosis or meiosis. And this is some pretty complicated stuff, and we're doing it very rapidly, so pay attention. Our objective is that you're going to use a model to illustrate the role of mitosis and meiosis in producing and maintaining complex organisms, and that's the objective. And it's very simplistic sounding because this is a lot of material about how cells divide and grow and why we need that to happen. To start with, all organisms reproduce in two ways. There's two types of reproduction, and the first one is called asexual reproduction, and the second is called sexual reproduction. Now, you may think, because of what we see around, we see all kinds of things that are sexual reproducers, animals and plants. You'd think that would be the most common, but by far, the most common type of reproduction is asexual reproduction. That's because for every human being on Earth, there are something like 100 billion bacteria, or some insane number. And bacteria reproduce asexually. Plus, when a cell in your body grows and divides, like a skin cell or a blood cell, we consider that asexual reproduction too. So, sexual reproduction is incredibly rare, but it does produce the things that are most visible, the plants and the animals. Okay, and there's a reason for that. But we're going to start with asexual reproduction. Unicellular organisms, which means single-celled organisms, such as bacteria, they generally reproduce asexually. Now, plants such as strawberries can also reproduce asexually. And there's other kinds of asexual reproduction as well. It requires only one parent. It creates a genetically identical offspring. Okay. Now, this can be advantageous because it's rapid. It's easy. The organism doesn't have to spend time trying to find a suitable mate. You can do it whenever you like, and asexual reproducers tend to reproduce at a very high rate of speed. I read one time that if you had enough nutrition, a single bacteria could grow to something the size of the planet. A colony could grow to something the size of the planet Earth in 48 hours. That's how fast these things can reproduce. Now, there's reasons that it doesn't, obviously, but asexual reproduction has a few advantages. But one of the main problems with it is that it's a disadvantage. That's why a lot of your large organisms, the sexual reproducers, are the more advanced organisms. Okay, because you don't get genetic variation. All of the offspring are the same, and genetic variation leads to genetic supremacy. And so as organisms have diversity, like two of the same parents produce different offspring. I have a brother who from the same parent, but we're very different. We're a lot alike, but we're very different, and all of that is happening because you have genetic variation in sexual reproduction. In asexual reproduction, you don't. In asexual reproduction, all of the offspring are the same as the parent, so the entire colony is genetically the same. This reduces your disease resistance. This reduces your ability to adapt to changes in the environment and can lead to massive amounts of die-off. So asexual reproduction has advantages. It's fast. You don't have to find another parent, but it's got some disadvantages too, in that the offspring are all identical, which makes them more susceptible. There are 5 kinds, some of which actually do happen in the larger organisms. We'll talk about that. But the first one is called mitosis, and we're going to spend a lot of time talking about mitosis today. Mitosis is basically when any eukaryotic cell divides. Now, eukaryotic cells are the ones that have a nucleus. There are cells that don't have a nucleus. They're called prokaryotes. But the eukaryotic cells have a nucleus, and mitosis actually refers to the division of the nucleus. When the entire cell divides, it's called cytokinesis, and the entire process is called the cell cycle. And yeah, we're going to go over that. But mitosis is the division of the nucleus. Well, if you don't have a nucleus, you can't divide. So mitosis only occurs in eukaryotes. Here's the other end of it. Binary fission is what happens to all those little bitty cells, the prokaryotes, that don't have a nucleus. Their DNA is just kind of laying around, and it's a much simpler form of split. But notice in both cases, one cell splits and becomes 2 cells. That's called binary fission. That's called asexual reproduction. In binary fission, a bacteria splits and becomes 2 cells. In mitosis, a eukaryotic cell, this could be a blood cell or a skin cell in you, splits and becomes 2 cells. This has happened a million times in your body since I started this talk. Another type is called budding. That's when a little bit larger organism, like a plant, or this is some kind of a sea creature, a little bud grows off the side, and that bud breaks off and becomes a whole new creature. You may have seen the episode of SpongeBob. This is called a hydra, by the way, because natural sponges do the same thing. And so what happens is just a chunk of the body breaks off and becomes a whole new creature, but they're genetically identical. In all of these, the offspring are genetically identical, and so there's no genetic variation here. Okay, this is how you plant potatoes. Like this potato here, you've seen this happen at the house. You just cut this thing in half and put it into the ground, and these eyes will grow and create a potato plant. Now this is vegetative propagation. It does happen, as you can see, in the larger organisms sometimes. These plants do have the ability to reproduce sexually, but we don't want them to. Agriculturally speaking, we would rather do it this way because we know we get the same result every time. But the entire field will then more than likely be genetically similar. And if there's a bacteria or a virus or something that comes along, it can kill one of them, it can kill all of them. And that's one of the problems with asexual reproduction. Finally, there's regeneration, and this is very interesting. There are starfish farmers down in the South Pacific because people eat starfish down there. Well, what they'll do is they'll catch about half the starfish in their little area that they hunt, and they'll take the other half and grab them and break them in half and throw them back in the water. And one starfish broken in half will become 2 new starfish. This is called regeneration. Lots of lower animals can do this, and so it is a thing that it's another type of asexual reproduction. The smaller, or the 2 new starfish are genetically identical. So one of the problems with this is that it does not create genetic variation, which is crucial to the long-term survival of species. Short-term, though, this is the way to do it because it increases the population very rapidly. So 2 goals of any species, whether it's a bacteria or a human, that is to reproduce rapidly and to be able to survive long-term. And those are kind of opposites of each other in terms of plans. So the really smart species are the ones that can do both. So of course, sexual reproduction involves 2 parents, and these 2 parents both have their own DNA, and they pass along a portion of that DNA to the offspring. And so when these 2 birds produce an egg and that egg hatches, it will have some of the genes of each parent. This produces an entirely new combination of genes, more than likely that's never been seen before, which gives it an opportunity, the new traits, the new characteristics, to survive the different world, the environment. And this is how most of the today's species have become so successful, is through sexual reproduction, which creates variation in the offspring. And variation in the offspring helps you to survive long term. Here's something to be clear about. Nature doesn't care about one organism, not at all. What nature cares is this species of organisms capable of surviving a change in the environment. When one organism dies, it's usually for the better for all of the other organisms, because that organism was not well suited to have the right traits to survive in that environment. Therefore, when it comes time to reproduce and create the next generation of organisms, that organism that died, its traits are gone, and those are the traits that we didn't want because it didn't work in the new environment. Okay, so sexual reproduction creates entirely new genetic combinations, and it creates organisms that have a much greater chance at surviving some type of change in the environment, which is, for the long term, a good thing. For the short term, it's more difficult because there's a lot of competition. These 2 birds had to find each other. The male probably had to fight off a few other males just to get the opportunity to reproduce, and it took a lot of time and energy, way more than just splitting in half like a bacteria does. So that's the disadvantage. It is much more slow. Sexual reproducing organisms, and there's some ways that there are organisms that beat that, but sexually reproducing organisms tend to reproduce much more slowly. Flowering plants are interesting because, first of all, flowers are an exact representation of sexual reproduction. There were no flowers. The whole purpose of this flower is to sexually reproduce, and there were no flowers before there were insects, but that's another story. Many plants pollinate, and that's how they reproduce, but some plants, in fact quite a few plants, can actually reproduce through something called vegetative propagation, which is you don't take the flower. It's kind of like budding. You take another part of the plant and you move it and plant it. We showed you the potato a minute ago, well, other plants are the same way. You can literally, some trees, you can just cut a branch off and go jam it in the ground and it'll grow. That's called vegetative propagation. There are some worms, planarians, annelids, some black worms. They do the same thing. There's these annelida worms that you can cut in half in the right spot and they'll grow into 2 new worms. It's crazy. Also, some wasps, ants, and fleas can reproduce even with only having a female. The female can go through something called parthenogenesis, which is they can actually create a viable zygote, which will become a fetus, which will become a living thing without being fertilized by a male. And so this parthenogenesis can create offspring without a male present. It's a little weird. And, again, it has the problems of not having the genetic diversity that you get from sexual reproduction. But if there's, if you're down to just a few females, this could keep the species going. So let's go back to asexual reproduction and talk about a very simple process called cell division. Well, it's not that simple, but it's not that bad. But there are 2 main types of cell division. The first is called mitosis. We're going to spend some time on that, and in a little while we'll talk about meiosis. Mitosis happens in almost every cell of your body. If you get cut and you bleed, to replace that blood, there are cells in your body that go through mitosis. A single cell inside of your bone marrow starts to go through this process, and when it's all said and done, there is a bone marrow cell left inside of your bone, and then another cell becomes a blood cell. This is how we create blood, and we create literally billions of blood cells an hour in our body. Okay, so this process is called mitosis. Well, actually, the process is called cell division, and there's 2 parts. Mitosis is when the nucleus on the inside splits. You can see it splitting right here. And notice that right here I've got 2 new nuclei. They've split. Mitosis is over. But I'm still technically having 1 cell. Finally splitting that cell down the middle to here is also, it's called cytokinesis, and it's the rest of the cell division. Okay. We're going to talk about the entire life cycle of this cell, including this very brief part of it. And we're going to basically talk about how 1 cell becomes 2, and then those 2 become 4, etc. One of the main things that happens during mitosis is that inside of this nucleus there are chromosomes, and one of the goals here is that you've got to get the exact same number of chromosomes here as here. So we're taking 1 cell and cloning it, okay? But I got to have the exact same number of chromosomes that I started out with in both cells, so at some point the chromosomes have to be copied, right? So let's look at the entire life cycle of the cell. This is called the cell cycle. This is how every cell in your body lives that's not involved in making sex cells, that's sperm or eggs. Okay, we're going to start right here. Now there's no reason to say, actually no, we're going to start right here. Actually, no, we're going to start right there. There's no reason to say that this is the beginning of the cell life, okay? But a lot of people like to start here because when you get right here to the end of this M, you've actually got 2 new cells. So let's start here with a brand new bouncing baby cell. And it goes through a very long point in its life called the G1 phase, or cell growth, okay? And that's all it does, is it grows. And it grows like crazy. It's growing like crazy in preparation to do this right here, which is to spend a lot of energy to copy those chromosomes. When you say, right now when you see DNA, you can think chromosomes. We'll talk about the difference later. So we spend some time right here. This cell is absorbing nutrition and it's growing. Its internal processors are working like crazy to increase its mass, and then it all of a sudden stops right here and starts copying the DNA. There's no more growth right here, but all the DNA is copied. You've got 3 billion pairs of DNA, and if you're a human, you got 3 billion pairs of DNA, and you got to make a copy of all those. So that takes a while. If you did it 1 pair per second, it would take you 75 years. Well, we do this in minutes, not years, so this has to happen very rapidly. And by the way, it has to be accurate. If you were 99% accurate on this, which is outstanding, I'll take it, when I'm doing other things, you would still have over 3 million errors in your DNA when you got done. You can't have that. So DNA duplication takes a long time, or the cell stops everything and spends all of its time on DNA duplication. And don't forget, it has to check it to make sure that it's okay. So we'll talk about these checkpoints here in a minute. After the duplication is over, there's another growth phase. We call that G2. And in the second growth phase, the cell is simply growing larger in preparation to divide, which is this yellow area right here. So let's pause here on the yellow area and go back and tell you this. Everything in here that's not the yellow area is called interphase. Okay, that's when the cell grows, DNA divides, and the cell grows some more. Okay, that's interphase G1, S, and G2. This area here, and you can see that even though this isn't really a timeline, mitosis is actually even slower than this, or faster than this in terms of the cycle of the cell. The M phase does happen in order every time, and there's 4 parts to the M phase, or actually 5 parts to the M phase: prophase, metaphase, anaphase, telophase, and cytokinesis. And at the end of cytokinesis, you got 2 cells. One goes back in, they both go back into this cycle. One goes this way, one goes this way, but both the same. And you know, 1 cell becomes 2, 2 becomes 4, 4 becomes 8, and that's how you do all kinds of things in your body. Every cell in your body does this. Well, nope, that's not true. There are a few cells that don't, and I'll explain that later. But almost every cell in your body does this. When you get cut and you lose blood, you have to replace that blood, and to do that, you grow new cells through this process. The skin where you got cut, the way that it fixes is it grows new skin cells right there, and then they connect together. That's what happens underneath a scab. And that's all done through the cell cycle. The skin is growing and dividing and growing and dividing until it fills the gap. This is an incredibly important part. One thing that I want you to notice is that it has to be done right, especially this duplication of DNA. If you screw up this DNA just a little bit, then you got yourself a problem, and the cell can usually just die. Therefore, there's a couple of spots where the cell stops and says, hold on, are we even ready to do this yet? The G1 checkpoint happens before the S phase, because the S phase is so crucial to be done right. So the G1 stops, or the cell cycle stops here at the G1 checkpoint and says, okay, everybody, hold on a minute. Let's all look around and make sure we're good. And then it starts the duplication. And then, before it splits in half, it does the G2 checkpoint again, checking to make sure that everything's right and ready to go. Plus, inside of the S phase, there's all kinds of little tricks that it has to make sure that it either doesn't screw up or is able to fix the screw-ups before it goes on. Okay, so this is actually fairly complicated. So again, just a quick review in the G1 phase. So this is where we started, like on the last slide. This is the starting point, the brand new bouncing baby cell right here. In the G1 phase, it enters immediately after cell division. It starts growing and functioning normally. This is where it does its job for you, okay? Because you know, not only cells don't only just grow, they grow and do a job for the body, okay? Skin cells, blood cells, hair cells. So this is where it actually does the work. The S phase is where the DNA is copied, okay, which is very important. The G2 phase is more growth, and then we go into mitosis, right? And notice this, what's happening here. We got 1 cell to start with right here, and at the end we have a brand new bouncing 2 baby cells right here. We want to talk about what happens during each phase of mitosis. So drawing a line right here to right here, this is all the M phase right here. So it's everything except for this first one. This first one is where everything else happens. Now this happens to be at G2 because G2 is right before prophase. But G1, S, and G2 happened here, and this one, and then when these are done, they go back to G1, S, and G2, and it happens again. So we've already talked about interphase. I'm going to skip that, but by clicking on these names, you get a big description. So I want to tell you what's going on during each phase, and yes, this is very important, so please pay attention. The DNA during the interphase is all open. It's fluffed up like cotton. If you were to look at it in a microscope, you couldn't even hardly see it because it's all it's very thin, and it's all fluffed out and ready to be used inside of the nucleus. But we're about to do something crazy here, and we can't just move that around like that. We got to literally pack it up. Now think about moving. I don't know if you've ever moved, but you don't just go grab all your stuff and throw it in the back of the car, because you're probably going to damage it. Either you or more than likely your mother or grandmother says, no, we got to pack that up. I know that a couple of times that I've moved in my life, my wife has made us pack things incredibly well, especially our good dishes. So I want you to think of the DNA as like your good dishes. You don't just throw them in the back of the truck and hope they don't break. because that would be, in this case, it would cause the cell to die. If the DNA breaks, the cell dies, right? So we're going to take care of it and pack it up. So the first thing that happens, the prophase is the packing phase, okay? All of the DNA starts to coil up. I'm going to link a video to that inside of Schoology. All the DNA starts to coil up, and it coils up and packs up into what we call chromosomes. So that's the difference in the two. DNA is unpacked genetic information. Chromosomes are when all that genetic information is packed up. Now there's an interesting thing. When the dishes are in the cabinet, I can use them. They're unpacked. But when they're packed up like this, I can't use them. And this is how I basically run the cell. So the problem is we don't want to have these packed up for very long. Just like I don't want my dishes packed up for very long because I need to eat. So we want to pack these up, and then we're going to move them, and then we're going to unpack them pretty rapidly. That's why the mitosis part, or the M phase, does not take very long. So prophase is all about packing. The DNA thickens up into chromosomes, okay? And they're attached because there was a copy made in the S1, so there's 2 copies of each. All right? And as I'll explain in a minute, there's actually 4 copies of each because you start out with 1 from mom, 1 from dad. We make a copy of each one of those, and now I've got 2 copies from mom and 2 copies from dad for a total of 4 copies. I know, just hang on, we'll teach you that in a minute. But each of the 4 copies they get packed up right here in prophase. Now in metaphase, we start moving, all right? So the first thing we got to do in metaphase is, here's the thing, we're splitting these between 2 cells. Now I got to make sure that every cell gets 1 copy from mom and 1 copy from dad. So I've got 4 copies here, all right? 1 from mom, 1 from dad, and 1 from mom, 1 from dad on each side. And notice how they're kind of lined up like this. Now here's 1 from mom, here's 1 from mom, here's 1 from dad, here's 1 from dad. Okay, so each one of these has 2 from mom, 2 from dad kind of thing. And these things right here, they're called centromeres. They act like cranes, and these little lines, they reach out and they grab a hold of these things. I'm sorry, these are the centromeres, these are the centrioles. They grab a hold of these centrioles, send out these little lines called spindle fibers, and they grab these at the centromere, and there's one coming from both sides, right? And so once everybody's lined up, and that's another checkpoint, by the way, this thing pauses right here and says, okay, is everybody connected? And if this spindle fiber has to be connected here and here from both sides. That's during metaphase. They get lined up, okay, and all these fibers connect, and we're ready to go. Then during anaphase, we're actually moving away from the center towards the edges, okay? These fibers start to contract and they start to pull, and they actually pull them apart, kind of like a wishbone, just pull them apart. Now this is 1 copy from mom here, 1 copy from mom here, and you'll notice they're heading in different directions. Here's a 1 copy from dad here, 1 copy from dad here, heading in different directions. Here's another copy from mom here, and another copy from mom, heading in different directions. When this is all said and done, this process actually puts the 2 copies from dad and the 2 copies from mom here, and another 2 copies from dad and 2 copies from mom here. I said that wrong. When this is done, it puts 1 copy from mom and dad here and 1 copy from mom and dad here, and it works. And this has happened, like I said, in your body thousands, possibly millions of times since you started this video, and we're on 23 minutes. So 23 minutes ago, in the last 23 minutes, this has happened in your body millions of times, and it's got to be done right. And if it's not done right, then the cell dies, which is merciful. It's a good thing. But when these things get pulled apart, and I get exactly 1 copy from mom and dad here and 1 copy from mom and dad here, that's the goal, and it has to be done right every time. So this structure is very critical. That's anaphase. And we finally reach telophase, and telophase is when we're through with the nucleus, okay? And notice, are the chromosomes there anymore? No, they're starting to unpack back into the DNA all fluffed out. That's called chromatin. And notice that the cell is starting to split up too. The nucleus is basically done. At the end of telophase, the nucleus is split up. And now the cell is going to split up, and the next part is called cytokinesis. And then we're done. But at the end of cytokinesis, I've created 2 new cells. I've got 1 copy from mom and dad here, 1 copy from mom and dad here, and now this copy is just like the original copy up here. Okay? Now, one thing we got to know is that that copying did not happen. You know how I've got 1 from mom and dad here and 1 from here? I started out with 1 from mom and dad, and I had to make a copy, so I now have 2 from mom and 2 from dad. I know this is getting confusing, but that copying did not happen during the M phase. That copying happened back in the S phase. And you got to remember that, all right? So that's how mitosis works. I need you to go through here and just kind of know basically what happens. Prophase, we're packing up. Metaphase, we're getting ready to move, so we're lining up. Anaphase, we're moving away from the center of the cell. We call this the equator, right? And then in telophase, we are, I don't have a thing for T, but in telophase, it's the nucleus is done, the DNA goes, it unpacks from the chromosomes, and the cell starts to split, and then finally cytokinesis. I've got an entirely split down the middle cell, and these become two new cells. This happens in your blood, your brain, your skin, your bones, your stomach. It happens all over the place. This is how we grow. This is how we heal. This is how we develop. This is a very crucial process happening in your body. Now we talked about these checkpoints. I told you about two. There's a third one right in the middle of M. Okay, but these checkpoints are, they have a specific job. What I wanted to talk to you about again is that the first checkpoint, the G1 checkpoint, sometimes the cell gets to the G1 checkpoint and there are signals from the outside or the inside of the cell saying, you know what, we don't need to go any further, we're going to stop. And they flip out into this G0 phase, and this G0 phase is a phase of rest. The cell goes into a resting phase. It does not have to grow anymore. Your skin's not constantly growing. So your skin is not constantly growing, it goes into the G0 phase and you stop. Now something happens. You get scratched, or just by scratching your skin, skin cells come off. You got to replace those skin cells. Now they come out of G0 phase and back into G1 and start the S process all over. But they'll pop out of G1 and pause until they need to go back into G1 and finish the thing. So that pause could be literally years, or it could just be minutes. It just depends on the need of the cell, right? Now this process right here, this G1 checkpoint and flipping into the G0, that is important when we get to talking about cancer later on, because this is where cancer starts, right here. This is a mix up in this G1 checkpoint, a screw up right here is what causes cancer. Okay? Don't forget the G2 checkpoint right before mitosis, and there's an M checkpoint in metaphase, right? Metaphase is when they're all lining up. That's the second phase. And remember how I told you that those fibers had to be connected, right? Well, there's a checkpoint there to make sure that those, here they're saying microtubules, but spindle fibers, microtubules, roughly the same thing, that they're connected right. If they're not connected right, then it stops until they fix it. So these checkpoints are really important, and they really help the cell from not screwing this process up, because it's complicated all the way around, and it needs to stop every once in a while and say, hey, okay, how are we doing? Did we do this right? If not, let's fix it. This is frightening to me because this is just one page dedicated to stem cells, but stem cells and understanding stem cells, your life is going to be so affected by stem cells. Stem cells are what we call undifferentiated, meaning that they could become any cell in your body. If you think about it, when you were first conceived, an egg from your mom and a sperm from your dad came together to create that first cell, and every cell in your body came from that. All of your skin, hair, blood, muscle, top of your head to the bottom of your feet cells, all came from that first cell. Well, that first cell then had to have the ability to become all those things. It's called undifferentiated, meaning that it has the ability still to become anything. When they become differentiated, there's changes that goes on in the cells, and they can't become anything else. Like muscle cells can't become skin cells anymore because they've already differentiated. So we're interested in stem cells. Stem cells have an incredible ability to heal. I mean, if we could get stem cells for you and put them in the right spot, we could almost heal any disease that you have. It's crazy. I have a friend who is in a wheelchair. If we could get some stem cells in the right spot, we could fix the break in his spinal column, and he could, through therapy, start walking again. And I firmly believe that's going to happen one day. But the problem with stem cells is right now the research is done on aborted fetuses. And there are people who have ethical problems with that. There are people who say we should not take an aborted human and do research on it. And so we are working on other sources of stem cells. We're getting better at it. We're finding stem cells that come from other places besides that ethically questionable place. And so we're already seeing people have stem cell therapy with incredible results. And within the next 20 years, you guys will probably see people you know getting stem cell therapy. Within the next 50 years, you will probably yourself have stem cell therapy of some form to fix something going on in your life. Stem cells are incredible. They also have an incredible ability to get ethically taken out of control. So something to know. There are two types of stem cells. The embryonic are the ones I was talking about that come from an aborted fetus. They get them from a fertility clinic. They are the best stem cells. They're the ones that give us the best ability to investigate. The problem is again, the moral or the ethical quandary. We're getting past that. We're just figuring out other ways to get this. You can actually now get this from embryos that live. So in fact, I was offered when my daughter was born, and she's 15 years old now, but I was offered to be able to save some of her embryonic stem cells. It was very expensive and I didn't choose to do it, but had I done that and they kept them in, the expense comes from keeping them in a cryogenic freezer. When she was 80 years old, if we could harvest, if she had the need, we could take some of those stem cells and they could use it to help regenerate any things going on in her body. They could literally grow her a new kidney if they needed to. Well, that was expensive, and I had been reading, and I know that we can actually get stem cells from adults now. So inside of you there are still some stem cells, and they may not be totally undifferentiated, they may be slightly differentiated, but we can find them in places like in your hip, right? This is your hip. We can find stem cells, and if we pull some of those out, grow them in a petri dish outside or some other type of container, we can actually then put them back into like a break in your bone, and we can heal your bones faster, or cure osteoporosis, or any number of other things we could do. Fix things like bone cancer, and that's just one example. So stem cells are pretty amazing, and we're just now we're no longer at the infant stage. We know a lot about them, but we're still at the kind of toddler phase. We are still learning an awful lot about how to use stem cells for what they're going to be. But in your lifetime, you're going to see stem cells become a huge influence in medicine. It will change everything. Okay, and now for the process of meiosis. Now, mitosis is when one cell splits and becomes two new cells and they're identical. Meiosis is a little different, and meiosis is only involved in sexual reproduction. Okay? Now, sexual reproduction is when I produce a cell that is going to combine with another cell from another parent to produce an offspring. I do not want all of my DNA in those cells. I don't. What I want is to contribute half of my DNA, and the other parent contribute the other half, and two halves make a whole. So the offspring creates a whole, it gets a whole set of DNA. If I send just a fraction more than half of my DNA, it'll more than likely kill the offspring. If I send a fraction less than half of my DNA, it'll kill the offspring. I want to send precisely half of my DNA to the offspring. The thing is with all of my DNA, think of it like a deck of cards, okay? With all of my DNA, and I only send half, which half do I send? And it's just a shuffle. The shuffling happens during meiosis. In meiosis, what actually ends up in the cell that is going to fertilize the egg, or if you're a female, what actually ends up in the fertilized egg is an absolute random split of your DNA. So imagine taking a deck of cards, thoroughly shuffling them, and taking exactly 26 cards off the top and putting it to the side. Guess what? That is exactly how meiosis works. It is random. That stack that you put to the side, that is the randomness that happens during meiosis. So we end up with cells that have a random combination of our DNA, and that's a good thing, because now your offspring, my offspring, whoever's offspring, this happens in plants and animals and all kinds of things, the offspring is an unbelievably diverse result of the mixing of two parents. Two humans could literally reproduce hundreds of millions, possibly billions of times, and never get the same combination. That's how crazy sexual reproduction is in terms of creating genetically different offspring. Alright, let's talk about meiosis. It's a two-step process, and it kind of looks like mitosis twice. Here I have a cell, okay? The DNA is copied, right? That's during the S phase. So far it looks just like mitosis. Then through a round that looks just like mitosis, prophase, metaphase, anaphase, telophase, it splits into 2 cells. These are called daughter cells. Well, these daughter cells go through another round of kind of like mitosis, prophase, metaphase, anaphase, and split again. And now instead of 1 cell becoming 2, 1 cell becomes 2, then 4. And instead of having 1 cell create 2 cells that have the exact amount of DNA, it creates 4 cells that have half the amount of DNA. And so now each one of these could become the sperm or the egg that gets fertilized. And notice the combination here. Notice how you've got red and blue chromosomes over here, and over here I've got, red and blue combinations, and then over here I got some red and some blue, but none of these combinations are exactly the same. That's what happens in sexual reproduction. We produce egg and sperm that have an unbelievably random amount of our DNA. But it is exactly half. It's just whichever half. Think about cutting a deck of cards in half. You don't know which 26 cards you're going to get in each one of these. Okay? I'm going to leave it to you to click on these and read these. I want you to know that these are basically prophase, metaphase, anaphase, and telophase, the same thing. Prophase, the DNA packs up into chromosomes. Metaphase, they line up at the equator. Anaphase, they start moving away from the equator. And in telophase, the cell splits in half. Okay? Now, then each one cell does it again. Prophase 2, metaphase 2, anaphase. But the same thing happens. They line up. Okay? And all that's really happening here is we're just ensuring that the DNA gets split in half and gets split in half again, and when we get to the end we got 4 cells, each having half of the DNA of the parent. Well, there's something interesting that happens in prophase 1 that I wanted to cover. It's called crossing over. This, let's say that this pink color here is the mom. One side of this is a full set of a chromosome. Remember, it's now copied during the S phase, so we have another side of it, right? So now I've got 2 full sets of chromosome from mom, and I got 2 full sets from my dad. Now, let me be clear. This is inside of me, for example. This is my mom's chromosome. I got a copy of it. Now I got 2. This is my dad's chromosome. I got a copy of it, and I got 2. Okay? And so when I go to pass this on to my offspring, basically one of these 4 copies is going to end up. My daughter or my child is going to get one of these sets of chromosomes from my mom or my dad. That's how sexual reproduction works. But here's the interesting thing. These things are so similar that when they're next to each other in prophase, they could swap like a little piece of their foot. And so now look, instead of having just all purple or pink, whatever color that is, or all blue, now this one did come from my mom, but it's got a little bit of dad's DNA on the bottom. And this one did come from my dad, but it's got a little bit of mom's DNA on the bottom. That's called crossing over. And it took us forever to figure out how this works back in the 40s. But we finally understood that that had to happen to explain some of the way that the DNA was transmitted. Okay? And the thing is this is yet even more genetic diversity, because now what would have happened was you'd have just got a chromosome from dad. But now I get a chromosome from dad with a little bit of mom's on it. So it gives me a chance of having an even more diversity of offspring. It is unbelievably crazy how diverse the offspring can be just from this sexual reproduction. And that that diversity of offspring is a really good thing. So this is why this is a good thing, this crossing over. It creates even new genetic combinations that we've never seen before. Okay? But I do want you to click through the rest of these, and I want you to see what happens during each. This is meiosis 1. And this is meiosis 2. These are the 2 cells that were produced at the end of meiosis 1. They go through the whole process again. And when you get to the end, you get 4 sex cells. These are either sperm or egg, and they have half of the DNA from the parent. Remember that the parent had 1 copy from mom, 1 copy from dad? Well, here I have half the DNA of the parent, and this is a copy, but it's random. Did this one come from mom, or did this one come from the dad? Which would ultimately be the grandparent for whatever this becomes. You're going to have to look through this a few times. I've tried to explain it as simply as I could. It's not that confusing. Maybe rewatch this, but understand basically that we're getting 4 cells instead of 2 in meiosis, and that each cell contains half of the DNA from the parents to be passed along to the offspring. So we are looking at 3 generations here. Parents, or here's the parent, but the parents' parents, if that makes sense. And then the offspring. So we're looking at grandparents, parents, offspring. And what we're talking about here is it's random which one of the grandparents this DNA, this chromosome gets, or this offspring gets. Alright, I know that got a little confusing, but hopefully you'll be able to get it. There's one other kind of interesting thing that happens in plants, and that is that after meiosis, okay, and it's called alternation of generations. I'm really not sure why it calls alternations. Well, I guess because it goes from a sporophyte to a gametophyte back and forth. But the sex cells actually multiply themselves to create this is in plants, to create kind of a spore body called a gametophyte, and that contains a bunch of cells. So normally in other things, like with humans and sperm and egg, the sperm don't reproduce through mitosis to create a whole bunch more sperm after they're produced. You know, you make a sperm, the sperm then goes and does its thing. But plants, you know, and again, the whole goal here is to create as many spores as they can. So each one of these spores is created after meiosis in a plant, right? These are the sex cells. They'll actually grow and make a huge body of these sex cells. It's called a gametophyte. And it's released. I don't know if you've ever seen pollen in the air. This is pollen, right? And this pollen gets everywhere. But that's what the plant wants. So the goal here really is just to make a bunch of sex cells. And then they just throw those sex cells out, like Mardi Gras beads, and they're just flung everywhere. And they go all through the air, hopefully gonna, find another sex cell on a plant, on a tree or a plant or something, and combine. Or the bee comes along, or the hummingbird comes along and gets these things. But this is interesting. The whole point is the alternation of generations is that after meiosis, and we've produced our 4 cells, each of those cells then goes through mitosis again. And that's the weird thing. That doesn't normally happen in almost anything except some plants. Plants, especially ferns, will do this. The sex cell will go through mitosis, and that's called alternation of generations. There's a video on it in Schoology for you to watch in the calendar. And I think the video does a better job of explaining than this. You're welcome to look through this. This talks about the 2 kinds, the spore forming, the sporophyte, and the gametophytes. It gets a little confusing, but just understand that mainly what happens is that right here in the sporangium, the sex cells are going through mitosis, not just the normal cells. And so it's kind of weird, but it's not that weird. And that's it. That ends this. And I'm just letting you guys know that normally I spend about 3 weeks to a month on this. So you need to really go back and look at this and pay a little bit of a special attention. Look at the supplemental material that I've included inside of the calendar, and make sure that you're doing all these things, because this is a complicated thing. Call me, text me, email me, Zoom with me if you have any questions, and I can try to explain this a little bit better. Okay? Mitosis and meiosis are not confusing, but you got to focus and pay attention to what's going on. Just look at each step and see what happens during each step. Alright, good luck.